everyone, and thanks for joining BioTeach again for this video on inhibitors. The information in this video will be useful for A-level biology and probably BTEC applied science to some extent too. So when we think about the word inhibitors or to inhibit, we know it means to stop or to hinder. Enzyme activity can basically be prevented or stopped by enzyme inhibitors. These are molecules that bind to the enzyme, which they then inhibit. There are two types of enzyme inhibition. There's competitive inhibition, which compete directly with the substrate for the active site, and there's non-competitive inhibition. This type of inhibitor does not occupy the active site. Instead, it binds to another area of the enzyme and distorts the shape of the active site, and as the shape of the active site changes, it means that the substrate cannot bind. The diagram here shows you an illustration of a competitive inhibitor. You've got the enzyme there in the shaded shape, and you've got two substrates, and you've got the inhibitor in the middle. Competitive inhibitor molecules have a similar shape to the substrate molecule, so they actually compete with the actual substrate to bind to the active site of the enzyme, but no reaction occurs. Instead, the competitive inhibitor blocks the active site of the enzyme, so the real substrate cannot fit. This means no reaction can occur and no products are made. Something to be aware of is that the competitive inhibitor effect can be overcome by increasing the substrate concentration. This graph shows substrate concentration on the x-axis and the rate of reaction on the y-axis. You can see the maximum rate of reaction there too, that's represented by the dotted line. Now, you have to note that if you increase the concentration of the substrate, then the reaction rate will increase. This is because you're making more substrate molecules available than the inhibitor, so there is a higher likelihood of an enzyme colliding with a substrate than with an inhibitor. I also think an important point to note is that if there's a high concentration of inhibitor, then nearly all of the active sites will be filled by that particular inhibitor, preventing the substrate to bind, and hence the reaction will slow or stop. Non-competitive inhibitor molecules bind to the enzyme but not at the active site. The diagram here illustrates this point. When this non-competitive inhibitor, shown on the left, binds to the enzyme, it distorts the bonds in the tertiary structure, and it changes the shape of the active site, making it impossible for the substrate to join. Basically, the substrate no longer fits, and so an enzyme-substrate complex cannot form, and the rate of reaction slows or stops. These inhibitors are called non-competitive, as they do not compete with the substrate. The graph here shows you the effect of increasing substrate concentration. The key thing here is to know that the effect of non-competitive inhibition cannot be overcome by increasing the substrate concentration. This is because the shape of the active sites have been altered, so adding more substrate will not make a difference. Increasing the concentration of the substrate will therefore have no effect on the rate of the reaction. Okay, so that's all I've got for you guys on this particular topic. I know it's short, but that's all really the main things that you need to know about the competitive and the non-competitive inhibitors. My next video will be based on required practical 1, and then I'm going to cover some past paper questions and share them on my test shop as well. Thank you so much for watching this BioTeach video on competitive and non-competitive inhibitors. See you soon.